So this is chapter 4, looking at evolution and the origin of the species. How did we come up with the different species? So in the Galapagos Islands, Darwin discovered that plants and animals were found there that did not occur anywhere else. And speciation is the origin of a new species, and this is the focal point of evolutionary theory. So evolutionary theory must explain how we get new species, how they originate, and how they evolve. Microevolution is going to be looking at changes in the allele frequency in a population over time. It's looking at it more on the genetic level, where macroevolution is going to refer to the broad patterns of evolutionary changes that you see above the species level. So the biological species concept emphasizes reproductive isolation. Species is a Latin word meaning kind or appearance. So biologists compare the morphology, which is the structure, physiology, biochemistry, and the DNA sequence when grouping organisms. What exactly makes a species is not really a fully agreed upon definition by all scientists. So the biological species concept states that a species is a group of populations whose members have the potential to interbreed in nature and produce viable, fertile offspring. They do not breed successfully with members of other populations. So gene flow between populations holds a species together genetically. So this automatically has a difficulty in dealing with the bacterial species that are going to do binary fission for their cell division. They don't actually breed with another member. So what we see is there is similarity between different species, but we also see diversity within a species. So all of these people are humans, they are all the same species, but they look slightly different. So reproductive isolation is the existence of biological factors or barriers that impede species from producing viable, fertile offspring. So the hybrids are the offspring of crosses between different species, and reproductive isolation can be classified whether these factors act before or after fertilization occurs. So reproductive isolation is the existence of biological factors or barriers that will impede the species from producing viable, fertile offspring. And so these are some examples here of pre-zygotic barriers and post-zygotic barriers. So pre-zygotic barriers, habitat isolation. If they don't live close to each other, they're not likely to breed. Temporal isolation, if they have different breeding times, different behaviors. Mechanical, they just can't do the deed. Or gametic isolation, so the gametes aren't going to be compatible. Post-zygotic barriers would be reduced viability of the hybrids, or the hybrids break down over time. So those pre-zygotic barriers prevent fertilization from occurring. So the different things that can block them from attempting to mate, from a successful completion of the mating, or if the mating is successful, it can prevent fertilization. So habitat isolation, if the species rarely encounter each other or occupy different habitats, they're not going to mate with each other. So a water snake versus a land snake. Temporal isolation, so if they breed at different times of the day or different times of the year, not going to mix their genes. Behavioral isolation, so if their courtship rituals are different or have different unique behaviors, they're going to not mate with each other. So pre-zygotic barriers, mechanical isolation, and gametic isolation. So mechanical isolation, here you've got two snails and they have a sidedness to them, and if you don't have the complementary side, they don't get to mate. So the sperm and eggs of one species may not be able to fertilize the eggs of another species. So post-zygotic barriers prevent that hybrid from developing into a viable, fertile adult. So the reduced hybrid viability, fertility, and the hybrid breakdown. Occasionally you will see some that you can have them breed and you may be able to get one generation to breed, but they won't continue to breed, and that's what we call the hybrid breakdown. So these are those post-zygotic barriers. So here you can see with the donkey, horse, and mule, so you might be able to get one more generation, but you're not going to get multiple generations. Reduced viability, so the genes from the different parent species may interact and actually impair the hybrid's development or survival. So a lot of hybrids, they may be vigorous and they'll survive, but they end up being sterile. And then some first generation hybrids are fertile, but when they mate with each other, with either parent species, the offspring of the next generation is feeble or sterile. So there are some limitations to the biological species concept. It cannot be applied to fossils or asexual organisms, including all of the prokaryotes. The biological species concept emphasizes the absence of gene flow. However, you can have gene flow occur between distinct species. So, for example, grizzly bears and polar bears can actually mate to produce growler bears. So you can have gene flow between those two organisms. So we have the grizzly bear, the polar bear, and the hybrid growler bear. So other definitions of species concepts will emphasize the unity within a species rather than the separateness of the different species. So the morphological species concept defines a species by structural features. This can apply to both sexual and asexual species, but it relies on subjective criteria. The ecological species concept views a species in terms of its ecological niche. It applies to sexual and asexual species and emphasizes the role of disruptive selection. The phylogenetic species concept defines a species as the smallest group of individuals on a phylogenetic tree. This will apply to sexual and asexual species, but it can be difficult to determine the degree of difference required for a separate species. So speciation can take place with or without geographic separation. So it can occur as allopatric speciation or sympatric speciation. So here you've got allopatric speciation, where you've got these that will divide into two different types, or sympatric speciation, where you have a mix in both, but they are actually able to interact with each other here. Allopatric, they cannot interact with each other. So in allopatric speciation, gene flow is interrupted or reduced when the population is divided geographically into isolated subpopulations. So for example, your flightless cormorant of the Galapagos likely originated from a flying species on the mainland. The definition of barrier depends on the ability of a population to disperse. So for example, you can have a canyon create a barrier for small rodents, but it wouldn't be a barrier for the birds, coyotes, or pollen that are easily able to travel there. Separate populations may evolve independently through mutation, natural selection, and genetic drift. So allopatric speciation can also occur in nature. There's 15 pairs of sister species of snapping shrimp that are separated by the Isthmus of Panama. These species originated 9 to 13 million years ago. When the Isthmus of Panama was formed, it separated the Atlantic and Pacific waters. So now you can see that there is significant visible difference between 4 of these species. So regions with many geographic barriers will typically have more species than regions that have fewer barriers. Reproductive isolation between populations will generally increase as the distance between them increases. Reproductive barriers are intrinsic to the organisms themselves. Physical separation alone is not considered a biological barrier. So sympatric speciation, this is going to take place in geographically overlapping populations. So this can occur if sympatric speciation can occur if the gene flow is reduced by factors including polyploidy, sexual selection, and habitat differentiation. So polyploidy is the presence of extra sets of chromosomes due to accidents during cell division. It's much more common in plants than in animals. Polyploidy can produce new biological species in Sympatry with a single generation. One of the reasons this will actually be viable in plants is because of the potential for self-pollination. An autoploidy is an individual with more than 2 sets of chromosomes that's derived from a single species. So here you've got a cell that's diploid. The 2n number is 6. When there's the cell division error, now it becomes tetraploid, where they become 4n. It will undergo meiosis to make 2 gametes that are both 2n, and then when those gametes fuse, you have a new species that's actually 4n. So this is an autoploid speciation event. So instead of having a new species that's 2n, the new species is 4n. So an alloploidy is a species with multiple sets of chromosomes that's derived from different species. With sexual selection, it can drive sympatric speciation. The selection for mates of different colors has likely contributed to the speciation of the cichlid fish in Lake Victoria. So here, when you have under normal light, this is what the fish look like. Under monochromatic orange light, the fish, even though they're different, appear very similar. So the sympatric speciation can result from the appearance of new ecological niches. For example, the North American maggot fly can live on native hawthorn trees, but it can also live on the recently introduced apple trees. So in allopatric speciation, geographic isolation restricts the gene flow between populations. Reproductive isolation may then arise by natural selection, genetic drift, or sexual selection in the isolated populations. Even if contact is restored between the populations, interbreeding is prevented. So in sympatric speciation, a reproductive barrier isolates a subset of the population without geographic separation from the parent species. So this can result from polyploidy, natural selection, or sexual selection. So a hybrid zone is a region where members of different species mate and produce hybrids. Hybrids are the result of mating between species that have incomplete reproductive barriers. So a hybrid zone can occur in a single band where the adjacent species meet. For example, 2 species of a toad in the genus Bombina interbreed along a narrow hybrid zone. So here is where you have this hybrid zone. You've got this fire-bellied toad and this yellow-bellied toad, and they have their own zones, and then there is this hybrid zone where they overlap. Hybrids often have reduced fitness compared with the parent species, so that means they aren't going to survive as well. The distribution of hybrid zones can be more complex if the parent species are found in patches within the same region. So many questions remain concerning how long it takes for new species to form, or how many genes even need to be different for it to be a new species. So broad patterns in speciation can be studied using the fossil record, morphological data, or molecular data. The fossil record includes examples of species that appeared suddenly and then persisted essentially unchanged for some time and then apparently disappear. So Niles Eldridge and Stephen J. Gould coined this term punctuated equilibria. This describes periods of apparent stasis punctuated by sudden change. The punctuated equilibrium model contrasts with the model of gradual change in a species over time. So with the punctuated model, here you have one butterfly species that then speciates into 2, and they will just persist the way they are for a long time. With the gradual model, you can see here that gradually over time, they become more and more different to give the 2 different species. So a fundamental question of evolutionary biology persists. How many genes change when a new species forms? So if you look at the difference between humans and other animals, particularly things like the chimpanzee, there's very little difference in the genetics between us. It only takes a small change, really, to make a different species. But how small is that change? So depending on the species in question, speciation may require a change in a single gene or many genes. For example, if you look at the Japanese snails, the direction of the spiral shell affects mating, and that's controlled by just a single gene. And so that's the end of the section. This picture didn't have any significance other than he was cute.